The last thing I want you to look at before we jump into the actual physical and chemical methods of microbial control is a slide that orders for you roughly how resistant different things are to these techniques. And this is a really rough guide because different organisms are going to have different sensitivities. But what this should be considered as is how challenging are these things to remove from the environment? How much do we have to think about? Do we remove these? So things like prions and endospores are hard to remove. So if we need to remove these from the environment, we need to use the toughest things. We need to use high heat, like 1,000 degrees, or an actual flame, or we need to use some really harsh chemical. Whereas if we're trying to get rid of typical gram-positive bacteria or enveloped viruses, maybe we can use ethanol, we can use hand sanitizer, something really simple and really ineffective like that. So that's really the way to think about this, is these things are easy to get rid of. Really simple techniques can get rid of these or kill these or inactivate them. And these require high-tech or harsh materials. And that's what you're going to see in the coming slides. Is in a lot of cases I'll talk about yes or no the techniques are able to inactivate endospores. And what you should take away from that is if they can inactivate endospores, they can inactivate everything else also. That's a pretty good assumption you can make. If a technique can destroy an endospore or a prion, it's capable of destroying everything else. What you will not see in the slides is a discussion of prions. And that's just because prions are thankfully rare in the environment. The vast majority of people don't have any in them, thank goodness. And so far, if you sample the environment, you don't find any unless you're in a place with chronic wasting disease. And the West Coast doesn't have that yet. So endospores are what we're more concerned about for a lot of reasons. Some really, really nasty diseases are caused by bacterial endospores. So we definitely want to get rid of those to prevent those diseases, but also because once we're sure we've done this, we've also gotten rid of all these. Okay, physical methods are what we're going to look at first. I will skip over some of this in these videos. What you should be studying is what the methods are, and then when we would use them and when we wouldn't use them, or why we wouldn't use them. So basically, compare them to each other and think about, in different situations, what would you use and why. And last, as this says, if you can't find some of this specific information in the slides, if it's not there, that means it isn't necessary for you to know it. Okay, so you don't have to go looking for additional information beyond what's in the slides. If it's not there, you don't need it. Or at least it won't be on an exam or quiz I give you. So the first type of heat-related disinfection we look at is moist heat. And specifically, we're going to start with boiling water, but we're also going to look at the difference between moist heat and dry heat. And that's what we can look at here on this table. We can compare the time it takes to sterilize material at various temperatures if we compare moist heat to dry heat. And if you look at this one, say 121 Celsius, I mean, that's very hot. That would be like steam heating. Versus just hot, very hot air. Well, if it's moist heat, it can sterilize something in under 15 minutes in some cases, compared to 600 for dry heat. So why is it so much faster to use moist heat for sterilization or disinfection than dry heat? Why is moist heat so much faster? Well, that's because water carries more heat than air does. Water, for whatever reason, water molecules in air carry a lot of heat, and they can transfer that heat to other materials or to whatever they're touching. This is an important distinction. Heat and temperature are not the same thing. So I'm not saying that that water can get hotter than dry heat. That's not what I'm saying. I'm saying it can carry more heat. And you just have to take my word for that. That's a thing in physics and engineering. And that's what's going on. So that's why we see a lot of different technologies that use moist heat. They either use hot water under pressure or steam under pressure. And ultimately that means that we can use those technologies to do sterilization or disinfection at lower temperatures than we would need to get if we were using dry heat. So what moist heat does is coagulate proteins, or basically unfold them and make them stick together so that they don't work anymore. And that is enough to kill any cell. Boiling in particular is mostly used for disinfection. And we make a distinction between this and sterilization by thinking about endospores. Can boiling water inactivate endospores? So if you get water from some source and you want to drink it, but you know there are endospores in it, and you boil it, what will happen? Well, you'll kill the vegetative cells in a few minutes of boiling. You'll kill the growing cells, but it might take many hours of boiling to inactivate endospores. And that's why we wouldn't use boiling water to inactivate endospores. It's just too slow. And that's just because boiling water is limited to 100 degrees Celsius. At sea level, or anything close to sea level, it's around 100 degrees Celsius, and it literally cannot get hotter than that. And so at that temperature, it just takes a long time to inactivate endospores. But vegetative cells are easily destroyed by those temperatures. So what you will see is that boiling water is a very handy, very easily accessible method of disinfection, and we use it when we cook soups, because when the soup boils, it disinfects itself. And that's how I see it when I'm cooking. I am killing bacteria. You can see it however you want. Boiling does other things too. But most of the technologies we use for industrial or commercial disinfection don't use boiling. They use things like autoclaves or pasteurization, as we will see on the next slide. So come back to this table as you see the different temperatures that come up in the next few slides. And this will really hit home for you, the difference between moist heat and dry heat. So an autoclave using steam is able to do the sterilization that we need in a laboratory or in a clinic. A lot of the times an autoclave is the most effective tool we have for sterilizing anything. And what it does is it takes hot steam, so much hotter than 100 degrees, and it pressurizes it. And what this means is that you can heat up water way past 100 degrees Celsius because you keep it under such high pressure it won't boil. And the other thing about it is, under that pressure you can achieve really high temperatures that carry a lot of heat. So moist air or steam holds a lot more heat than dry air does. And if you remember back to chemistry or physics, you add a certain amount of heat to get water up to boiling temperature, but it takes a lot more heat, a lot more physical energy to actually boil the water. And so steam has all that extra energy in it, and when it touches a surface and condenses, it releases that energy into it. So that's why steam carries a lot more heat than the same temperature dry air would. And that, if we go back to this previous slide, that's why if you compare moist heat at 121 Celsius to dry heat, something that took 15 minutes to kill in an autoclave would take 10 hours to kill using just hot, dry air. And that is a thing. So yeah, autoclaves do this. And so what it's showing is a pressure vessel with a huge metal door that has to hold back this incredible pressure. And this piping, what it does is it introduces steam, and then it's able to pressurize it. Different autoclaves work in different ways, but what they all do is heat the steam and put it in this chamber at a high pressure. The other thing I'd point out about autoclaves is these are the temperatures autoclaves work at. So typical autoclaving of something like water, if you want to sterilize water or growth media in a laboratory, use 121 Celsius. An autoclave can typically also achieve a temperature like 134 Celsius, but not at a high enough pressure to safely do water. So if you want to autoclave like metal parts, you'd use this temperature. The tricky thing is that high pressure steam corrodes metals. And so anything you're going to autoclave, any metal you're going to autoclave, has to be stainless steel. And it turns out autoclaves are made of stainless steel. Yeah, and so that does kind of limit what you can use an autoclave for. So typically we would autoclave like glass, Pyrex containers. Those are not going to be damaged by the heat or by the steam. But you would never autoclave a computer if you spilled a bacterial culture on it, for example. The next physical method of moist heat is pasteurization, and this is what we use on food, liquid foods a lot of the time. There are three different types, broad types of pasteurization historically, and you don't have to memorize these numbers. What you have to do is compare them. So the first type of pasteurization involved heating a vat of something like milk to 63 Celsius. So 63 Celsius is not quite hot enough to burn a person, and it's not the 100-degree boiling temperature. So it's much less than that. And if we hold milk at that temperature for 30 minutes, it kills the majority of vegetative cells. So it kills the types of bacteria that would typically contaminate milk. And what that means is that milk will be a little more stable. It won't rot as quickly, and it won't be as dangerous. If somehow endospores got into milk, this would not affect them at all. But usually there are no endospores in milk. This is a slow way to pasteurize anything, and it's very energy intensive. So a newer, more common technique is what they would call flash pasteurization, and this is where they have liquids flowing through pipes. And that's what this is showing, is the liquid goes through pipes, and the pipes go through a hot section where everything in the pipe gets heated, and then they go through a cold section where it rapidly gets cooled again. And this allows us to keep things moving through the factory and control the temperatures. And so if the temperature goes up like 8 degrees, we can achieve the same pasteurization in a much smaller amount of time. And this would be less likely to affect the flavor also. And so again, with pasteurization, what we're trying to do is kill the vegetative cells that we would expect to contaminate milk or juices, but not sterilize them. If we want to sterilize them, that is, if we want to make them shelf stable and we want to make milk that we don't have to refrigerate, that is, we seal it and don't refrigerate it because there's nothing alive in it that could rot, we use ultra high temperature pasteurization, which uses a much higher than boiling temperature for a really short amount of time. So if you send milk through one of these systems under extremely high pressure so that it won't boil, and you get it up to 140 Celsius, this is higher than an autoclave temperature. If it's at that temperature for 6 seconds, it will inactivate endospores. That milk will be sterile, and that's what shelf-stable milk, how shelf-stable milk is made. We don't see this much in the US, but if you look at like a Parmalat box that would be more common in Western Europe, that's how they make it. These pasteurization techniques are for refrigerated milk. You don't have to sterilize it if you're going to keep it in a refrigerator, because even if there are a few endospores in it, they're not going to grow in the cold. And so pasteurization combines heat, which kills vegetative cells, with cold, which prevents growth. So heat is bactericidal, cold is bacteriostatic. So dry heat, as I've mentioned before, requires higher temperatures than moist heat. And we do use it in situations where water would damage whatever we're trying to disinfect. So we use dry heat a lot of times to sterilize metals that would corrode if they were exposed to water. And in a lot of cases, we try to use temperatures up closer to 200 Celsius and for something like an hour. And we're typically imagining something like an oven that we would put materials into. And what I want to point out is that even though I have kind of belabored the point that moist heat can be used at lower temperatures than dry heat, one form of dry heat is the gold standard for disinfection and sterilization, and that's incineration. Incineration is the best way we can sterilize a material. So if a material can survive being heated to something like 800 degrees Celsius, where it turns red hot, it is sterile. There is no biological material or anything that can survive that heat. All biological molecules, like the amino acids, nucleic acids, proteins, everything changes their chemical form at temperatures like that. Many of them will become carbon dioxide and just go away. Water boils so fast it ruptures every cell. So dry heat methods that get to incineration temperatures are completely effective at sterilization, far beyond anything else you see in this lecture. So keep that in mind. Incineration really is the gold standard, and they use this whenever possible because it is the most effective thing. So certain techniques we use, incinerators. So in the laboratory, we use inoculating loop, and you can get an electric incinerator where you can stick the loop in here and it will be heated to red hot in about 2 seconds. And that's hot enough to kill anything. It's hot enough to turn prions into carbon dioxide. So that's great. And you can get ovens that would do the same thing. Like you could have a sterilization oven for surgical tools or something like that. I don't actually know what they use on surgical tools. It's been a long time since I've read about that. But you could use this to sterilize metals that would be damaged by an autoclave, for example. But you would have to use the much higher temperatures, like 170 Celsius, and much longer times, like 2 hours, compared to 120 Celsius and 15 minutes in an autoclave. But yeah, incineration using an actual fire, that will sterilize anything. This is a slide for your curiosity if you want to compare different temperatures. These are some different things. So if you see this in the upper right corner of a slide, it means it's just for your curiosity, and I don't expect you to study it if you don't want to. Filtration is another physical method of microbial control that just separates the microorganisms from the rest of the material. And We would use this if we're doing something like trying to sterilize a solution of vitamins that would be destroyed by heat. We could send them through a filter. We would do this if we're trying to sterilize the air we breathe. We'd send it through an N95 filter. And this is much more relevant now that we are all dealing with COVID and everyone wears masks. And the idea with these is that they block the majority of microscopic particles. So 0.3 micrometers is smaller than most bacteria, bigger than most viruses. But that's kind of misleading. You don't really need to know the size of these because most bacteria and viruses would be in large water droplets, or at least much bigger than that. So when you exhale microorganisms, whether they're viruses or bacteria, they are in droplets of water that do get trapped by a typical mask. So if you see somebody telling you not to use a mask because of these numbers, ask them to think a little bit more. I mentioned cold talking about milk and food products. Cold in refrigeration is bacteriostatic. Deep freezing, so something like minus 80 Celsius is a deep freeze. It's a temperature you never encounter on Earth outside of Antarctica, but we have freezers that get to those temperatures in laboratories. We use them to store microorganisms in a bacteriostatic way. So we store them and we get them back out again when we want to use them. So you'd never use this as a sterilization tool. It's more like you would store microorganisms in a deep freeze if you wanted to use them later, or you'd store food in a refrigerator if you didn't want microorganisms to grow. Another physical method is ionizing radiation. This is one of the most effective ways of killing bacteria and endospores in foods. And the idea is we have different ways of making things like X-rays and gamma rays with extremely high energies, and those destroy DNA and proteins. So if you have a source of those rays and you pass food near that source, any microorganisms in the food will quickly be destroyed. And this is the symbol that irradiated food would have on it. Irradiated food has a bad reputation in the US just because anything involving radiation has a bad reputation in the US. But it's a great technique for protecting foods. And ultimately, they do check to make sure that the foods that get irradiated aren't made radioactive. There's no known health hazards associated with irradiating food. Compare the effects of this radiation to the effects of cooking. Radiation affects the food a lot less than cooking it does. And the other thing you could think about is compare the effects of radiation on food to the effects of pathogens that you might eat on your food. The radiation is a lot safer than something like salmonella. But it doesn't matter that much because a lot of the foods in the US are not irradiated. It's an uphill battle to get this adopted. What we do use more often is ultraviolet radiation. So this is just ultraviolet light, and it's able to damage DNA. So you can use this for disinfection. If you have a non-porous surface. So the insides of biosafety cabinets in laboratories are disinfected using ultraviolet radiation. But you can't use it for anything porous, like a sponge or a cloth that would block the light. So you can only damage cells that are directly exposed to the light. So that's enough for this video. This was the physical methods of controlling microbial populations. The next set of slides, or the next part of this lecture, is chemical methods. And that won't take as long. There aren't as many concepts to go through in the chemical methods.